4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 17. 18 means 3 are absent. Yeah, 2 are absent. Then the 2, 4, oh, there are 6. Oh, okay. Okay, so students, please come. What is today's date? Today is 18th. Today is 18th of Jestha 2081. But in 19th Jestha 2058, a tragic incident happened in Nepal. That means near about how many years ago? 23 years ago. No, 2058. Near about 23 years ago, there was royal massacre. Yeah, there was royal massacre, right? It was a Friday. What was it? Friday. So I think every Friday or on first Friday of every month, all the royal members, they used to gather together and they used to have gathering as well as party. But 14 persons, they were killed. Yes or no? King, queen, and his means crown prince, right? Crown princess. It means Virendra side all 5. Husband, wife, 2 sons, 1 daughter, and his means third brother also, right? And other sisters, relatives, so many were killed. And some they blame to somebody, some they blame to somebody, but who knows what is done. Anyway, afterwards, Nepal slowly changed into republic country. Up to that time, it was fully kingdom, right? Yeah, it was. There was active monarchs, and Nepal was only the country uh, whose religion was Hinduism. Only one country, but now no country has religion as Hinduism, right? Now there is secularism, right? There is secular state. Here is this one. Today also presentation. Okay, what is there? Presentation. Who are the presentator of today? Ritika, Luniva, Simres, Sasank, Nilisa, means 5 presentators are there, right? Joy Kumar, when did you do your presentation? Question answer. So he hasn't done presentation. At first we have to give chance to him, okay? Because it was there, his activity, but not presented means next time he will not do. And he will say, whether I prepare a presentation or not, it doesn't matter, so he will not ask me. That's why it's better to ask to him first, okay? Please come. Now, the question answers. Joy. Find out your documents and then present, right?
Namaskar everyone, I am going to discuss long question answer today. So first question is, why did Mendel select pea plant for his experiment? What characteristics of pea plant were chosen by Mendel for his experiment? And the reason why Mendel select pea plants for his experiment are given below. So number 1 is, a pea plant is naturally self-pollinating, and artificial cross-pollination can also be performed on it. This helps to maintain the purity of characters in offspring. Second is, it has a number of distinguishable contrasting characters such as tall and dwarf pea plant, red and white flower, round and wrinkled seeds, etc. Thus the result of experiment can be checked by observing different characters. 3 is there, hybrid plants obtained as a result of cross-pollination is in pea plant are also fertile in many cases. It is found that such offspring are sterile. 4th is, a large number of progeny can be obtained from a single plant. The life cycle is of short duration, so the result of experiment can be obtained in short period. The plant are convenient to handle, so they can be grown anywhere. And next question is, would Mendel have succeeded if he had chosen frog instead of pea plant for his experiment? And the answer is no, Mendel would not have succeeded if he had chosen frog instead of pea plant for his experiment because of following reason. The life cycle of frog is long, about 2 to 10 years. It is difficult to breed in frog as it only breed in rainy season. Frog do not have the same favorable genetics characteristics that made pea plant ideal for studying inheritance. See, what are dominant and recessive characteristics? Clarify your answer by giving example. In a cross, those traits which express themselves by covering the other characters are dominant characteristics. Example, tall is a dominant trait which is expressed by capital T. In cross, those traits which are presented but cannot express themselves due to presence of dominant trait are called recessive characteristics. Example, dwarf is represented by small t. Describe crossbreeding and selective breeding briefly. Selective breeding, also known as artificial selection, is a process by human to develop new organism with desirable characteristics. Selective breeding is one of the earliest forms of biotechnology, and it is responsible for many of the plants and animals that we know today. One of the earliest examples of selective breeding in domestic dog, which humans have been breeding for at least 14,000 years. In crossbreeding, mating of two animals of different breeds is done. Superior traits that result in the crossbreeding progeny from crossbreeding are called hybrid, visor, or hairy toys. Last question is, what is the role of different types of research? Explain briefly. Role of different types of research are forensic. DNA plays an crucial role in many criminal investigation with extraction from samples like hair, skin, or blood. Forensic teams use DNA to identify suspect or eliminate them from considering it can establish person's innocence or guilt and determine if they were present at the crime scene. Paternity test. Paternity test. DNA extraction is valuable in determining the parentage of child, whether confirming or refuting parenting claims. DNA from both the potential father and the baby provides evidence. Ancestry tracking. Beyond identifying immediate ancestors, DNA extraction helps individuals understand their ancestral origins. Modern DNA kits reveal countries of origin, living relation, and potential predispositions to medical or food conditions. Medical test for certain medical conditions. DNA extraction is essential for official diagnosis, especially when the condition is genetic. For example, include cystic fiber, Huntington disease, or Down syndrome. Genetic engineering. DNA extraction aids is generally engineering plants and animals. For example, for plants, it helps identify, isolate, and extract desired genetic genes for replication in success generation. In animal, DNA extraction is crucial for tasks ranging for cloning to transforming one animal's DNA to another. Thank you.
So, he has brought question answers, but one question I think it is incomplete. I found. Which one is that? Here is one question. Describe crossbreeding and selective breeding. So he wrote here about selective breeding only. Is here crossbreeding?
Yes.
Where is it?
Crossbreeding.
In crossbreeding,
Breeding of two animals.
breeding in crossbreeding,
In third last line.
third last.
Oh, so he has written, but it must be written in separate paragraphs. Separate paragraphs, right? Okay, so I hope you might have done all questions. Have you done? Yes or no?
Yes.
Completed or not? Very good. Now, who is first presenter of today? Nilisa Joshi. Because this exercise was less to explain, so I called Joy. Now it's the turn of Nilisa to present on her PowerPoint. What is there? Let's check.
Blood circulation in human.
Your introduction is gone and write
Yeah.
about circulation.
Namaskar, it's me Nilsa Joshi from grade 10 standing here to tell about blood circulation in human bodies. The flow of movement of blood throughout the body. Blood carries oxygen, nutrients and other important substance from the heart through the blood vessels to the rest of the body, cells, tissue and organs. Pulmonary circulation. Pulmonary circulation includes a vast network of arteries, veins, and lymphatic that functions to exchange blood and other tissues, fluid between the heart and the lungs Systematic circulation. Systematic circulation carries oxidized blood from left ventricles through the arteries to the capillaries in the tissues of the body. Thank you.
Thank you very much. Okay, so in Nilisa's department, there was blood circulation. What is blood circulation? Flow of blood from heart to different parts of the body and vice versa. What can you say? The flow of blood from heart to different parts of the body and vice versa, means from other parts of the body to heart. That is called blood circulation. So in blood circulation, the heart pumps the blood and the blood flows through arteries, reaches in cells and tissues with the help of arterioles, capillaries. Again it is collected by venules, veins and comes to heart. Okay? And how many types of blood circulation are there? Two types of blood circulation. What are the two types of blood circulation? Pulmonary circulation and systemic circulation. Cell circulation. What is it? It is the circulation of blood between heart and lungs. Heart pumps the blood, that blood reaches to lungs. From lungs, the blood again comes back to heart. Just these two organs. Actually, this is also very important. What happens? When the blood is pumped out from heart, it reaches to lungs and it gains oxygen. Then that blood becomes oxygenated. Carbon dioxide is thrown out. Exchange of gases takes place in lungs, and pure blood or oxygenated blood comes to left part of the heart. That is called pulmonary circulation. In pulmonary circulation, which blood vessels are involved? Pulmonary artery and pulmonary vein. Pulmonary artery and pulmonary vein. Pulmonary artery carries deoxygenated blood. Deoxygenated blood means where there is less amount of oxygen, a bit more amount of carbon dioxide to lungs, and pulmonary vein carries oxygenated blood from lungs to heart. Here we have to remember, we discussed already about three types of blood vessels: arteries, veins and capillaries. Arteries, they carry oxygenated blood except pulmonary artery. Make concept clear, eh? All arteries, they carry oxygenated blood, but pulmonary artery carries deoxygenated blood. In veins, They carry deoxygenated blood except pulmonary vein. Pulmonary vein carries oxygenated blood. Understood or not? Now here is systemic circulation. What is systemic circulation? The figure shows here. Here is heart. Can you see heart or not? From heart, the blood goes to lungs, comes back to heart. That is pulmonary circulation. From heart, blood goes out and reaches to different parts of the body, and now again comes back to heart. Flow of blood from heart to different parts of the body and again back to heart, but it does not go to lungs. That is called systemic circulation. Mainly how many types of circulation? What are they? Pulmonary circulation, systematic or systemic circulation. In which circulation more blood is supplied? Systemic or pulmonary circulation? In systemic, more blood is supplied because it has to cover wide area. Yes or no? In pulmonary, only less area, no? Heart to lungs, lungs to heart. Are they far away or nearby? In between the two lungs, there is a heart. Right lung, left lung, in the middle, heart. Thank you very much, Nilisha. Now it's the turn of next presentator. Simresh or Luniba? Luniba. Mananda. No, look, Linda Mananda. Good. Okay. Please pay attention as you say. Just a minute, just a minute. You make in the bigger, okay?
Namaskar and good afternoon to you all. As you all know, it's me, Ramananda, standing in front of you to give a presentation on the topic of heartbeat and pulse rate. The rhythmic contraction and relaxation of the ventricles of the heart maintain the heartbeat. The total number of heartbeats per minute is called heart rate. The normal heart rate is 60 to 100 per minute. The alteration in heart rate indicates abnormalities. The factors that increase heart rate are exercise, fear, anger, sickness, etc. Heart rate below 60 beats per minute is called bradycardia. Heart rate above 100 beats per minute are called tachycardia. The pressure of blood flowing in artery against the arterial wall creates pull. Normal pulse rate are 60 to 100 beats per minute. Blood pressure. The pressure exerted by the blood flow on the wall of arteries is called blood pressure. The pressure is maximum when it is systolic condition. The pressure is minimum in diastolic condition. The pressure is categorized into systolic pressure and diastolic pressure. Blood pressure. The pressure of blood on the artery when the ventricles are contracted is systolic pressure. It is maximum pressure on the wall of the arteries that ranges from 90 to 130 mm of Hg in healthy person. The pressure of blood on artery when the ventricles are relaxed is diastolic pressure. It is minimum pressure on the wall of arteries that ranges from 60 to 80 mm of Hg in healthy person. Raise in BP is high blood pressure and fall is low blood pressure. Thank you.
Okay, thank you, Luniba. So Luniba, she had brought about heartbeat, pulse rate, and blood pressure, right? So contraction and relaxation of the ventricles. Ventricles contract, relax, contract, relax, contract, relax. So that is connected with heartbeat. So how to define heartbeat? The rhythmic contraction and relaxation of ventricles is called heartbeat. Oh, your heartbeat is slow. They say. Oh, your heartbeat is more high. I think both are dangerous ones. Right? If the heart beats faster, so blood pressure is high. If heart beats slower, blood pressure is low. Isn't it? So normal heartbeat. What is the normal heartbeat? 60 to 100 per minute. It must beat 60 to 100 per minute. But generally 72 to 80 times in 1 minute. But when we do exercise, when our cricket player plays games, when he is in anger, in danger, at that time, heartbeat is high. But one thing we have to keep in mind, heartbeat is connected with our breathing. Fast breathing, more heartbeat, and slow breathing, slow heartbeat, isn't it? But if the person is suffering from high blood pressure or low blood pressure, it is not connected with that breathing also, because heart is not functioning well, no? Okay, an alternation in heartbeat, when the heartbeat alters, either increases or decreases, that is called abnormality. What do you call it?
Abnormality.
Abnormality. So, what are the two abnormalities? If heartbeat rate is less than 60 beat per minute, that is called bradycardia. Remember this word, bradycardia. If heart rate is more than 100 beat per minute, what is called tachycardia. Okay, tachycardia. Next one is the pressure exerted by the flow of blood on the walls of artery is called blood pressure. Have you measured your blood pressure? So blood pressure has got mainly two values, upper value and lower value. That value comes later on. So pressure is maximum in systole condition. When the ventricles contract, the blood exerts more pressure on the wall of artery. That is systolic pressure. When the ventricle relax, less pressure is exerted. That is called diastolic pressure. Systolic and diastolic. So two types of pressure are here. What are they? Systolic pressure and diastolic pressure. So last time what happened? I wanted to donate blood, right? So my blood group is O positive, and I think blood donation is very good. And before donating the blood, they measure our blood pressure, whether blood pressure is up to date or not. By measuring blood pressure, they can find out the functioning of heart also. And my blood pressure was how much? 90/60. It means my blood pressure was low. What was it? Low. Then which one is high? Which one is normal? This thing we have to remember. Before that, just learn here. I have marked here two pressure ornament. Upper value is here 90. Lower value is how much? 60. So this pressure is called systolic pressure. What is it called? Systolic pressure. And this value is called diastolic pressure. That means this pressure is caused due to contraction of ventricle, and this pressure is caused due to relaxation of ventricle. Contraction of ventricle, relaxation of ventricle. So my blood pressure was low. What is the normal blood pressure? Normal blood pressure 120 to 140, upper one, lower one 60 to 80 mm of G, millimeter of mercury. So for normal blood pressure, systolic pressure or upper pressure must be between 120 to 140. Diastolic pressure should be between 60 to 80. Susan's blood pressure is 130/50 mm of Hg. So what type of blood pressure has he got? Do you know? Upper one is 120 to 140, means it is in the range. But lower one is below this one, right? it means he is suffering from low blood pressure. What is this?
Low blood pressure.
Remember, eh? Upper is okay, but lower is not okay. That's why low blood pressure. Raju's blood pressure is 130 by 80 mm of Hg. So, what type of blood pressure has he got? High blood pressure. Why? Because upper value is more than 140, no? Lower is okay. Lower it can go up to 90 also, 60 to 90. Okay, this is the thing. Next one is systole. I just said that is by ventricles contraction. And what is given here? 92 on 30 mm is given, but 120 to 140 also is normal. And here diastolic, how much is here? 60 to 80. And what is the device to measure blood pressure? Which device is used to measure blood pressure? Children, tell me. Yeah, one pump means one thing is tied here. Then what is its name? That is called sphygmomanometer. What is it? Sphygmomanometer. Sphygmomanometer. Sphygmomanometer is the device that is used to measure blood pressure. But nowadays there is automatic blood pressure measuring device. Simply just give the finger and then after some time it shows the digits.
Yes.
Right? But when we use sphygmomanometer, we have to use stethoscope also. Stethoscope. Something is kept here and then measure it, right? High and low. Okay. Thank you very much, Luniba. Right? Simaris is ready.
No, my first.
Ritika is ready? Okay, it's the turn of Ritika Maharjan. Yes or no?